So once more again, good afternoon. And I would like firstly to thank in particular, Professor Mangini, but also his team members for organizing this Congress. And I hope that now you can see just the first slide, is right?
Yes.
So fine. So in fact, I was participating also on the first session, and I've seen there are many young colleagues with different backgrounds, from different universities, also different departments. So I think it would be fine that I will give some short introduction on this topic, because logically the time is limited. And I would like to speak about flavonoids. I think that most of you know many information of flavonoids, and they are abundantly present in different plants, but also they're a very important part of our diet. And if you look here on the first slide, generally the different categories of flavonoids are somehow differently localized in different foods. Like, for example, isoflavonoids are typically located in soya. The most or the richest compounds from this category are flavanols, which are, in fact, we are taking like 100 milligrams daily, and depending if you are eating more chocolate, coffee, wine, everywhere, you can find flavanols. Citrus fruit are typical flavanols, and berries are rich in anthocyanins. Flavonols are just presented in different classes of food. Some examples, apples, and flavones are those which are the less presented in our diet. Just you can see some, for example, some less common vegetables or also spices. Well, in my lecture, I would like to start with explanation why we can be interested in the effect of flavonoids in relation to the cardiovascular system. So this information are largely coming from epidemiological studies. And these are some examples. And in those studies, there was an analysis between the intake of flavonoids from diet, but this intake was measured by questionnaires, food questionnaires, and those persons in those studies were followed for different period of time. You've seen sometimes even 10 years, sometimes even more. And the outcome was whether they were suffering less or more from some cardiovascular diseases. In fact, what is very positive, according at least to these studies, if you are taking more flavonoids, you have lower risk to develop a cardiovascular disease. In this table, green means low risk, orange means a tendency, and red means nothing. So in fact, what was fine, there was no negative effect. In fact, if you was taking, or those people were taking more flavonoids, generally have not a risk of developing a cardiovascular disease. On the contrary. In fact, we should be careful with interpreting this data, because they were, as I told you, based on food questionnaires. And moreover, if you have seen where those flavonoids are presented. So if you are logically eating more vegetables, fruit, generally your health style is, or your common habits are healthier. So generally, this may be a confounder. Anyway, there are also some studies in humans and animals, but you will see they are not very sufficient, but still there are. Like here are example of some human studies. I should stress at the beginning that many of them were mixtures. So for a pharmacology like me, it's quite complicated to say what was the really active compound. And many of them were also supported by some companies, like chocolate companies, for example. So again, they can bring some confounders. But one, from my point of view, is important that soya, containing, as you have seen before, as I told you, isoflavonoids, and cocoa, rich in epicatechin, for these two food, in fact, and I hope that also for those flavonoids, we have quite good data that they have some effect. These are mild effect. Logically, they are not clinically used drugs, but as a food, they can have some positive effect. Like isoflavonoids can decrease blood pressure and lipids, and cocoa as well, somehow mildly blood pressure. If you look on animal studies, or again, we can speak a lot about those studies, but I would like to pay your attention to one model, which is isoprene model. Is a model of acute myocardial infarction, and in fact, I started my research as a PhD student at the time with this model. At the time, yeah, there were some studies showing that really flavonoids are very protective. But well, at that time also I was aware that it's very different if you are taking flavonoid orally, because you will see also today in my lecture, that is being very extensively metabolized. So therefore, we decided to give some real flavonoid intravenously in order to be sure that we are testing the effect of flavonoid. So we gave routine, or we selected this one because you can dissolve it in some biologically friendly solvent. In this case, we use sodium bicarbonate. But surprisingly, I'm not going to discuss all the graph, but this model has about 30% mortality, which is something relevant before we introduced those correct interventions, because this was the mortality on acute myocardial infarction before, and some compounds were protective. But as I'm speaking now about the flavonoids, you see that very surprisingly, the routine, the flavonoid, was the only one which deteriorated this model. So in fact, not 1/3, but 1/2 of animal diet. And well, this was quite surprising. So we decided to look more in detail on those previous studies which were performed. And just a very short comment, because I don't like to speak about this. You will see that probably this data were not really real data. They were probably fabricated. As you can see, this study from India, this should be ECG in animals. These are our ECG. So apparently, if you will give isoprenolone, you will cause huge damage to the heart, and really the ECG is very variable. You see that these are all ECG after isoprenolone. And those, I know that you are an expert on HPLC here, so you might think that rather HPLC than ECG. In fact, is something which is not real. So therefore, we decided to test the same which one of this group did. In fact, they found full protection with no protection. So, well, something was not real in those studies. And it was quite complicated, to say the truth, to publish this paper. It took 1 year because we got 6 pages of comments of reviewers. But after 2 years, we published this study. And you can again see that it's very huge variability, but always there is a damage to the heart, and no, it is quercetin is unable to protect the heart. And unfortunately, these are again ECG. I can skip it again, something which is not a real ECG. And this is not the only one study which published this. There is another study you can see in Molecules, in which those authors gave quercetin for 30 days in 3 different doses. And we recalculated data which they in fact published. And you see something which is not possible. Give quercetin for 30 days in 3 different doses, and they found in almost all cases very high linear relationship. In particular, if you understand a little bit ECG, so the first one, the A, is the amplitude of T wave, which is not possible to be so linearly correlated. In fact, we sent a comment to the editorial board, and they gave 2 months to authors to response to send an answer to us, but they didn't react, so they after published the comment. Well, it was something which I think is quite important also to show the younger colleagues that not always which was published is really based on real experimentation, or experimentation according to really scientific approaches. Well, but in the next lecture, I would like really concentrate finally to the science, not to this unpleasant results. And this is the molecular mechanism, how flavonoids, and you will see rather the metabolites of flavonoids can have some effect on the cardiovascular system. If you look historically, and I think all of us know it well, that flavonoids were considered to be antioxidant. They are antioxidant fact, but you will see that this is not so simple, and it could not explain the possible positive effect of flavonoids. So the theory is very clear. We have a balance in our organism between antioxidants, which are, for example, vitamins C, E, but also antioxidant enzymes, superoxidismutase catalase, and reactive oxygen-nitrogen species. I must tell that they are physiological. Yeah, this is not reactive oxygen-nitrogen species that are sometimes needed. For example, hydrogen peroxide is needed for synthesis of thyroid hormones. So there's not nothing which is pathological in general, but it can become pathological when there is an excess of those reactive oxygen-nitrogen species. And we are speaking about so-known oxidative stress, which is really following many diseases. Well, so the logical theory, if this happened, why not to give some antioxidants, which are flavonoids, for example? But we know well that all clinical trials with antioxidant failed. This is not related only to flavonoids, but all vitamin, which were also tested. Why? This you will see, or one of possible theory I will tell you also today. Another approach is to chelate metals, because copper and iron can, in fact, facilitate the production of those species. The reaction is very known, is so-called Fenton reaction, which is with ferrous ions and hydrogen peroxide, but it can work as well with other metals, like with cuprous ions. Yeah? And logically, if you will chelate the metal, you would expect to decrease the production. So therefore, it is another approach. And if you look on flavonoids, we did some testing with chelation of metals. This is standard clinically used chelator deferoxamine for comparison. And for our surprise at the time, we found out that one flavonoid, bichelane, was so potent as the clinically used iron chelator deferoxamine, which was very promising. But this was the beginning, in fact, because you must check that really this compound is able to decrease the production of hydroxyl. And I should stress that antioxidants, they are also able to reduce metals, so they can have the opposite effect, increase the production of hydroxyl. So in fact, we tested that. We showed that iron was reduced, so really this potential of flavonoids to reduce iron is presented there. And we look on the Fenton reaction, and deferoxamine, in fact, as a clinical use standard, really decrease the production of hydroxyl. This was quite clear. But if you look on flavonoids, we found very different curves. And there was a disappointment, because you have seen that bichelane was so active as the deferoxamine in a very simple chelation assay, but in this Fenton reaction, it was not able to reduce, or very mildly, the reaction. You see the deferoxamine is this brown curve here. So, well, these experiments were performed with, I would tell, low sophisticated equipment. So thereafter, we had troubles to test very high concentrations, in fact, high ratios. So therefore, in collaboration with the University of Bologna, with Professor Mercolini, we developed a new methodology, which enabled us testing in much higher concentration ratios. And we did the same with copper, but the results were again disappointing. So really, like bichelate in this case was pro-oxidant. In fact, it potentiated the Fenton reaction. So, well, one explanation of this failure of antioxidants really to relieve antioxidative stress, to really bring a clear profit in those diseases, that in fact you see that by giving antioxidants, sometimes you can also facilitate a reaction. So this is one explanation, but I think much more probable and important in the case not of other antioxidants, because this can be true, but in the case of flavonoids is that they have very low bioavailability. You have seen where those flavonoids are presented, so it's not important which type of flavonoids or which group of flavonoids taking, but in fact all of them are very extensively metabolized. And not only in the liver, just in the intestine, in the small intestine, there are really many enzymes, the Regent enzyme, in fact. Flavonoids are there immediately conjugated with, or to sulfate, glucuronic acid, or methylated as well. And even some of them effluxed out of the cells. So they are continuing their journey. And, well, some of them which are absorbed, they are still metabolized by similar enzyme in the liver. So therefore, the bioavailability is very low. But importantly, a very large portion of those flavonoids are reaching the colon. And the colon, they are metabolized by our microbiota into different phenolic acids. And this scheme is quite complicated, I know. I'm just not going to tell detail, but I'm just purportedly showing this scheme here. As the green here are flavonoids. In fact, we know that there are at least 4,000 flavonoids in the plants. So there are really many, but those phenolic acids, they are much less. And really, this scheme is showing that the same metabolite can be produced from different flavonoids. In fact, the testing of flavonoids metabolites is simpler, and it can explain us why a different class of flavonoids may have the same effect. So, but anyway, we are continuing with testing. Just these are very preliminary data, and really some of those flavonoid metabolites are also able to chelate, reduce metals, and also they have some real protective effect against, for example, these are assays in which tested the protection against erythrolysis caused by excessive copper, which is relevant, for example, in Wilson disease, which is a disease in which there is very high level of copper. Well, just shortly, the other possible way how to decrease the oxidative stress is to block those enzymes which are producing reactive oxygen-nitrogen species. And there are these three enzymes as far as we know. And in fact, flavonoids are able to block them, but well, the concentration again are probably not achievable. And in collaboration with my colleagues from University of Pécs in Hungary, we tested also some metabolites, but most of them are not active. So probably this is not the mechanism which really, can be responsible for protective effect. And in the last part, I just would like to dedicate the time to our probably the most important discoveries in this area, and these are related to antiplatelet effects and effect on vascular smooth muscle cells. In fact, if you look on literature, you find many different studies showing that flavonoids are able to affect many pathways. But again, pay attention, those concentrations are very high, so mostly these studies are not very relevant. Anyway, we look on this cascade, which is a central. You see the cyclogenase 1 thromboxane A2 thromboxane synthase, and you can very simply monitor this cascade. Yeah, you can buy, for example, recombinant enzyme. You can test whether those compounds are blocking this enzyme. You can use the platelets to test whether they can block thromboxane synthase. And look also on the thromboxane receptor. In this case, you need only a stable agonist, which is here this U46619. In fact, the study started casually as we had some coumarins, and those coumarins were potent inhibitors of cyclogenase 1. You can see even more potent than the known aspirin, acetylsalicylic acid. And it was a quite short step from coumarins to flavonoids. In fact, we found out that some flavonoids, but these are not many, but some of them were active, and isoflavonoids were even more potent inhibitors of cyclogenase 1. This was recombinant enzyme. We look also in the plasma because this may be influence. And in this case, acetylsalicylic acid was more active. But interestingly, those isoflavones were active in low micromolar concentration, which may have logically some clinical relevance because some low micromolar concentration are achievable. We look also another part of this cascade, thromboxane synthase. Yeah, some compounds were active, but this is not important because the concentration was very high, 100 micromoles, so this is not the mechanism of action. We look also on thromboxane receptor, and here some compounds were active. The concentration were a little bit higher, but here the interpretation is not so simple as we use some stable agonists in contrast to biological thromboxane A. So to sum up this effect, if you look on flavonoids, some of them, they are able to have an effect on both steps in the cascade. So they are able to block cyclogenase 1 as aspirin, acetylsalicylic acid, and some are also antagonists and thromboxane receptors. And these are isoflavonoids, which are having both effects. Some other flavonoids are having only the effect at thromboxane receptors. And really, we have also some chemical explanation for this phenomenon. As you can see, there's a thromboxane A2 and the thromboxane, really, if you look, there are some structural similarities to those active flavonoids. Well, as the isoflavonoids were the most active, we look on them, and we found out that one of those flavonoids, tactorigenin, was the most potent. You see, the potency was much higher than acetylsalicylic acid. We look more in detail, and we really confirmed that the effect was based on antagonism of thromboxane receptor. It's quite interesting, as in most countries we are not using drugs acting as a receptor, and they can enrich our palette of the compounds. But well, still we are not satisfied. Well, this is quite active, but still we are VDC50 about 16 micromoles, which is still a little bit more. So we look on those metabolites. There are some previous studies, but were semi-quantitative. So we look on other, and they are tested rather the metabolite, not metabolites, but those phenolic acid presented in plants. So we look on the metabolites. In fact, we look what is available, and we bought all flavonoid metabolites, those small phenolic acids and or small phenolic compounds, because not all of them are acids. And you see some of them were quite active at very high concentration, but most of them were not. We look also on the active one, and we found out that one of this compound, 4-metacatechol, was incredibly active. You see we are now at EC53 micromoles, and this is 10 times more potent, so 1 order more potent acetylsalicylic acid. We look on the mechanism. In contrast to acetylsalicylic acid, there was no inhibition of cyclooxygenase 1. There was some thromboxane inhibition, but again at high concentration, so this was again not considered to be relevant. And there was no antagonism at thromboxane receptors. So apparently the mechanism was different. So we looked more further. We confirmed firstly the result of collagen, which is the physiologically most known or most physiological inducer of fat aggregation, and again confirmed like the 1 more order potency between methyl catechol and acetylsalicylic acid. And also in collaboration with my colleagues from University of Vienna, we confirmed this effect in vivo-like model, which is, you can see here, this is embryonic heart hen egg model. And really, if you will stimulate aggregation, you will, within 24 hours kill, you see that 60% of those embryos, but acetylsalicylic acid and methyl catechol, even particle, was almost inhibiting all mortality. Well, if you look here, these are the different cascade and the platelets, and what you have seen, methyl catechol is blocking collagen-based pathway, arachidonic acid-based pathway, has no effect on cyclooxygenase 1, and mild effect on thromboxane synthase, no effect on thromboxane receptors. So we're at this point a little bit lost because we need to look what is the mechanism, and major pathways were not blocked. So we thought that maybe it can somehow interfere with thromboxane A2, because there are no data how the thromboxane, which is a very unstable compound, is able to stimulate its extracellular receptors. So we look on that, but no, this case was not true. In fact, there was no effect of intracellular level of thromboxane A2. So we look further, and we found out there's some cascade by which the calcium is elevated. You see there is a catechol, so catechol can potentially chelate metals. So we tested firstly whether this is true, but no, in fact, addition of metals did not, maybe the exception iron, but it was probably peroxidation, did not affect the antiplatelet effect of this compound. So we decided as whether it can interfere with calcium signaling, and in fact we used a very simple compound which is binding calcium and transporting it inside the cells, platelets, and really you see that methyl catechol was active. So in some way the compound interfered with calcium signaling. Logically, these data were published, but we are not satisfied because apparently the mechanism was not very clear. So we look on different pathways. I will skip it now. There's a new paper which I hope will be published soon, as we are in the revision phase. And just to summarize it, we look on other cascade, and finally we found out that still the effect must be somewhere here. It's calcium relevant, but somewhere here. So what we did a very simple experiment, whether even if it's not blocking this enzyme, can somehow block the coupling, collaboration of these two enzymes. So very simple experiments. We gave the substrate, arachidonic acid, we measured the final product thromboxane A2. In fact, really it seems that is a major mechanism. As you see here, that acetylsalicylic acid is in this assay 10 times less potent, which seems to be relevant for the final effect. This is the summary. I think skip it, and I can tell you that now we are recruiting patients. These are the first results, which are with healthy donors, and really healthy donors. This compound is more active than salicylic acid, at least at the equivalent doses or concentrations. And the last part of my talk would be about different model, but still we are speaking about the same compounds more or less. Effect on vascular smooth muscle cells. And this study started after we read this interesting paper published in 2012. And this study was interesting as the authors gave quercetin by oral gavage, so by oral way, by intraperitoneal way. In fact, they achieved more or less the same level of conjugates in the plasma, but surprisingly, the effect of oral administration was much higher. And at that time, we were thinking that, well, probably those phenolic metabolites formed by microbiota can contribute to this. So we look which metabolites were available again, and we tested all of them on a rat aorta to cause whether they are able to relax this aorta. In fact, we found out, that many of them are very active, some inactive, but one was very active. You see again just difference between more or less one order. So logically, what we did after, we gave the compound by intravenous administration to rats, and you see that really there was a dose-dependent decrease blood pressure. Also, we mimic the real situation, as this is a metabolite, so it means that it's slowly being produced by microbiota, slowly absorbed. So we gave the compound by intravenous infusion, slow infusion, and really there was some dose relationship and a decrease in blood pressure. But surprisingly, even if we continue infusion, after 5 minutes, the blood pressure returned to the control values. It was something which we didn't understand at the time, but was rather disappointing because this was speaking against our theory. So we look also on mechanism, and we found that generally the most active compounds was endothelium dependent, but other compounds are having different effects. I'm not going into detail as the time is going to the end, so we can discuss it later if somebody is interested in, but there were some differences, and therefore we decided to give them together. And we combine all of them together, there were 3 compounds, and combination of the most active compound, which you have seen, the metabolite, with 4 metacatechol, in fact, the same compound, which is also able to cause block pathway degradation. And this combination, you see that really the drop in blood pressure remained. So this is probably the reason that really, in this case, not one metabolite, but a mixture of the metabolites is responsible for the effect. And well, the time has gone, I think 25 minutes is more or less precisely. So just I would like to conclude, and I hope that really I showed you that flavonoids are a possible source of novel drugs. So it's something related to sustainability. But I think that really not the flavonoids itself, because they are really poorly absorbed, but rather the metabolites are responsible for the effect. So I would like to thank all my collaborators, because logically this work wouldn't be possible to perform with my collaborators, not only at the faculty, but also my collaborators with other universities. And with a look on the current picture of our group, I thank you for attention. Logically, I would like to hear some of your questions.